Okay, great. Welcome everybody to the MIDAS webinar series. Today we're going to hear from recipients of the MIDAS COVID-19 Urgent Grant Program Awards. We have 4 presenters today. My name is Harry Hockheiser. I am an associate professor of biomedical informatics working with Wilbert Van Panwees in the MIDAS Coordinating Center. I'm also at the University of Pittsburgh, right down the street from Wilbert. And I'm going to be your host today. So without going into too much further ado, I'd like to get started and get to our panelists, our presenters here. Our first talk is going to be from Elizabeth Carlton. Dr. Carlton is associate professor in the Department of Environmental and Occupational Health at the Colorado School of Public Health. Done some work in environmental epidemiology and has a lot of interesting work on genomics and field epi, including schistomiasis elimination, which sounds like something I'd like to learn about, having been in a spot where that was prevalent. So without much further ado, let me hand it over to Dr. Carlton, and looking forward to hearing your talk.
Thank you. Thanks for the invitation to be here today. Put my slides. So I'm going to talk to you about a project we're working on trying to understand the role of climate and specifically meteorological conditions in the transmission of SARS-CoV-2. This work is really the work of a team of scientists, Jude Bayham at the University of Colorado State University, who's an economist, Andrea Buchwald, infectious disease modeler at the Colorado School of Public Health, Elise Grover, my doctoral student in environmental and occupational health, and David Jacobson, who's a data scientist at Vanadata. And our work is funded by the Colorado Department of Health and Environment, which I'll talk about in a second. But the MIDAS support that we have received has really given us the breathing room to dive into what I think is this really interesting topic as we all try to respond to this pandemic. So this question of the role of meteorological conditions in the spread of SARS-CoV-2 is nested within a larger project that's been taken on by the Colorado COVID Modeling Group. We're a group of social scientists, epidemiologists, infectious disease modelers, biostatisticians who are advising the state health department and elected officials in Colorado on the course of SARS-CoV-2 in the state. So we generate weekly modeling reports trying to estimate what's happening now and what may be happening in the future. The question we have been interested in as the focus of this talk is how does weather impact the spread of SARS-CoV-2? There's been a lot of really nice work done on this already on the coronavirus and prior to the SARS-CoV-2 pandemic on influenza and other respiratory viruses. For the purpose of brevity, I'm going to focus on kind of the pathways that we have been thinking about, which are really 2. If weather impacts the spread of SARS-CoV-2, we posit that there's 2 possible pathways by which this may happen. One is that weather may impact the environmental suitability for transmission. Certain weather just may make virus more transmissible or the respiratory tract more susceptible to infection. And then the second component of this is behavioral, that weather may influence the amount of time people spend indoors or outdoors, either in high-risk environments or away from high-risk environments. If we think about this in terms of beta, in terms of the transmission rate, you could really think about environmental suitability being about transmission probability. If there is a contact, what's the the extent to which weather is impacting the probability that a contact leads to a successful infection. On the other hand, the behavioral component really impacts, we think, may impact the contact rate, right? What is the probability that people are interacting with others in a way that might spread infections? So I'll walk you through some preliminary results here and our thinking on this topic. This is the course of, sorry, I'll start by just saying, why do we think this question is important? Well, this is the course of SARS-CoV-2 in Colorado in terms of the daily number of patients hospitalized with COVID-19. This is hospital census data, which I argue is a fairly sensitive measure of the state of infections in the state. As you can see in Colorado, we've had really 3 waves. The early wave that just about everyone experienced, we had a summer wave in infections, and then this real blast of hospitalizations peaking in December. And we're now at this point in mid-March where hospitalizations are oscillating. And I will say at each of these peaks, we were faced with this really challenging question of what might happen and what might be the impact of policy measures on the course of SARS-CoV-2. As you know, almost everyone throughout the US experienced some form of lockdown in summer, right around this wave, this peak. Statewide mask order was implemented, and around here, the state implemented what we call a red dial, which was a targeted lockdown in the state. And the big question right now is, given the ramp up in vaccinations, what may happen in the months ahead if policy measures are lifted? And I think one of the questions in the back of a lot of people's, in the back or the front of a lot of people's minds is how might seasonality of SARS-CoV-2 influence these policy measures, either relaxation or maintenance of policies in the days and months ahead. You know, one of the big open questions is, were we able to get away with a certain amount of behavior in the summer because SARS-CoV-2 was or was not impacted by meteorological conditions that we could not get away with in the winter peak? And this plot here on the left just shows where we think we are in terms of the uncertainty of where we're headed, which is to say there's a large degree of uncertainty. And to the extent to which we could understand how meteorological conditions are influencing transmission, it may allow us at least to tease out the role of weather from potential changes in policy or education or behavior. So this is our conceptual framework. Here you have outdoor temperature and relative humidity and absolute humidity. These are 3 meteorological conditions we're thinking about. Pathway 1 is that outdoor climate essentially influences indoor climate. This is somewhat straightforward to many of us. And that may lead these indoor environments to be more or less suitable to SARS-CoV-2 transmission. The other pathway is this outdoor environment influences how we spend our time, which can lead to transmission. It's worth noting, of course, there are other levers at play that may more significantly or more impactfully modify transmission, such as changes in policy or a real education risk perception that may change behaviors such as mask wearing or hand washing or other measures. We're doing this work in 5 counties in Colorado, 3 in the Denver metro region. This is Denver, this is Boulder, and this is Douglas County. They represent a diversity of the Denver metro region. This is El Paso County, where Colorado Springs is located. And lastly, we're including one of our mountain resort communities, which is up in Western Colorado. So to answer this question about environmental suitability, we have used purple air data to look at the relationship between indoor meteorological conditions and outdoor meteorological conditions. Many of us became familiar with purple air, those of us who live in the West, when the wildfires were burning this summer. They're most known for their provision of PM 2.5 measures, but they also record temperature and humidity. And so you can use these paired data, and the metadata indicates whether indoors or outdoors to understand, to look at the relationship between indoor and outdoor conditions. I think it's worth noting this focus on indoor conditions in that people spend about 90% of their time indoors, which you'll see. And we think these indoor environments are much higher risk for SARS-CoV-2. In fact, there are very few documented instances of spread of SARS-CoV-2 outdoors. This is perhaps the most obvious finding that, temperature indoors follows a much more narrow range than temperature outdoors, although it is correlated. Relative humidity is not well correlated indoors and outdoors. To any building scientists, this is probably quite obvious. However, absolute humidity is, in that absolute humidity, as outdoor absolute humidity increases, so too does indoor. And this may be important because there's some really nice studies showing that absolute humidity may influence transmission probabilities. So pathway 2, behavioral. What is the relationship between indoor meteorological conditions and time spent indoors away from home? So we used mobile device data combined with publicly available data on building footprints to estimate the time spent at home, outdoors, away from home, indoors, away from home, and traveling. And I'll just spend a minute on this plot because I think it's quite interesting. This shows in blue time at home. It includes both time indoors and outdoors at home. So if you're in your backyard, you're still classified as at home. In green, we have the time commuting or moving around biking. And then in orange, we have time outdoors, not at home. And then lastly, we have in dark orange or in red, we have time indoors, not at home. You can see the profound effects of the lockdown starting in March on how we spend our time. You can see these acute seasonal or weather events impacting time. We had a big snowstorm here, and actually we're about to see the impact of another big snowstorm we had in Colorado. But I think what is notable is these week-to-week oscillations that you see, which I can show you in this quick preliminary results plot, is that one of the biggest drivers of how much time you're spending indoors away from home, not surprisingly, is whether or not it's a weekend or a holiday. In fact, you know, perhaps the biggest driver of how we spend our time away from home is work, right? We do see a positive correlation between temperature, no matter how we slice and dice our data in different sensitivity analyses. And we're still trying to unpack whether this relationship between humidity and time indoors away from home is robust. There is some evidence for a seasonal effect in that we see more time spent at home, indoors away from home in the winter. So just a couple of key takeaways, and I know I'm at time. SARS-CoV-2 transmission appears to be limited outdoors. And so if outdoor meteorological conditions influence the spread of SARS-CoV-2, it's likely through changes in behavior and or indoor environments. Key driver of time indoors away from home is likely work or whether or not it's a weekend holiday. And lastly, we're seeing some evidence that the outdoor environment may influence the indoor environment in ways that may be relevant to transmission probabilities. So we're just kind of halfway through these analyses and hope to have more to share in the months ahead, hopefully in the form of a publication. So thank you for the invitation to talk today. Sorry, this is a little bit rushed given the time constraints, and I'm happy to take any questions.
Great, thank you. Any questions for Dr. Carlton? So I'll throw one out here. Is it sounds like there could be a pretty good opportunity for generalizing these methods for future efforts. Could you speak to that?
Yeah, we decided to take a deep dive into 5 counties because we wanted to do this as proof of concept. And we have hospitalization data at the county level and actually at the age stratified and for Colorado. So we were able to do it here, but I think the idea is this could be replicated on a larger spatial scale. And as many of you know, the environmental conditions in Colorado are quite different than other regions of the United States, right? In terms of we're kind of a dry, elevated area. So yes, we were very interested in kind of running this as a proof of concept and then seeing if we could roll this out at a larger scale.
Okay, and I think we have 2 other questions here that we can go through quickly. One is asking if you have any equations that you publish about the effects of temperature and humidity. Is that something you're going to be putting out there to
yes, we are hoping, okay. In the end, what we're hoping to do is be able to parameterize this for inclusion in a model. Nice. To be continued, but yes, that is okay. The goal.
And then the other question being, what are you using for mobility data?
This is a great question. So we had been using SafeGraph, and we are transitioning, and the data I showed you is from Xbox that Vanadata helps us to process. But great, it's all cell phone-based mobility pings.
Great, thank you so much.
Thanks so much.
Okay, so moving along here, Our next presentation is going to come from Kyle Dahlin. I hope I got that right. Who is a postdoc at the University of Georgia, who's interested in using models to describe the transmission dynamics of infectious disease, and particularly looking at zoonotic risk and wildlife disease systems, which I don't think you're going to be talking about today. And I think what Kyle is likely to talk to us about today is how institutions and the policies thereby affect or are affected by pandemics and other outbreaks. So Kyle, please take it away.
Thanks. Maybe there are quite a few similarities between universities and wildlife populations, but I won't be talking about that today. So this study that I'm going to talk to you about today is motivated by, during this pandemic, we saw that universities and other large organizations had to set their own policies to protect their members from infection and transmission of SARS-CoV-2. And perhaps in recent memory, that's not something that's been left to organizations. You've had more top-down policies that were set. So we decided we wanted to take a kind of retrospective analysis of what happened in the fall. So we're probably familiar with the different ways that universities and other organizations responded to outbreaks. Generalized interventions, of course. We saw in the spring that many universities moved to remote operations, putting their classes online, having their faculty and staff work remotely. But as we moved into the summer, COVID tests became more abundant and accessible. We saw universities plan to implement testing policies for either symptomatic or asymptomatic students and other individuals on campus, as well as some enforcement measures like isolating cases that they've detected or quarantining contact traced individuals or asymptomatic individuals. And the decision to implement some or all of these responses were largely guided by models of varying levels of sophistication. We started with very simple models, and in these simple models you get in general threshold behavior. Basically, if you can reduce R0 less than 1, then you'll have very small outbreaks. It'll just be, whoever, any imported cases won't infect any new individuals on average. And so with these models predict this behavior here in green, where if you can control, perhaps through testing or moving enough people off campus, enough beyond this threshold, you should have outbreak sizes close to 0, whereas you just have this decreasing behavior below the threshold. An alternative hypothesis is that you just get a simple, perhaps linear or other strictly decreasing relationship between your outbreak size, however you define it, and control intensity. So we're kind of curious whether or not this model predicted behavior matches at all qualitatively with what was observed in the fall. So we started out by formulating some simple models of transmission in university communities that incorporate different control measures that were used. So in this top right area, we have a simple model that incorporates testing as a way to take latently infected and asymptomatic infected individuals and move them to an isolation compartment, as well as a campus community transmission model that incorporates kind of a metapopulation model with on-campus and on-campus populations that also has commuting and travel. So for instance, if you don't move everyone off campus entirely, you might have restricted the number of days per week that people work on campus or the number of hours per day that they're there. But you still tend to have, even campuses that moved fully online, residential communities that live on campus, et cetera. And with these models, we're able to determine thresholds defined by the control reproduction numbers that kind of give us an idea of how much you would have to institute such policies in order to suppress transmission. So what I've described so far has been the first two parts of the plan for this study. And after receiving this grant, we moved on to data collection, and we're not really at the analysis yet. So we were able to bring a statistician on board, and I'm not a statistician, so I'm greatly appreciative of being able to bring them on board and begin this data collection process. So we're collecting data kind of from two different areas. One, how universities responded in terms of their testing plans, how they enforce those, generalized interventions, how effective they might have been, and whether how far they've moved online or remained on campus. And there's some data published prior to the fall about what universities plan to do. And Davidson College is putting together a nice database that tells us what colleges actually did in the fall. And we're working with them to get more information about that. In terms of case numbers, the New York Times began collecting data on on-campus case numbers, or they began reporting it October 8th of 2020. And that data, from our knowledge, includes all the cases that campuses reported from the start of the pandemic, so around March. And they continue to publish that data on a month-by-month basis. And we're somewhat able to determine outbreak sizes between October 8th and December 11th, which is a portion of the fall semester. And then we'll add to that some community level case data from the CDC and counties. I'll share with you some preliminary data that just uses the plans that universities had prior to the start of the fall semester, and this limited case data from the New York Times that's just covering October 8th to December 11th. So one question is whether or not having a testing plan in general lowered case numbers in any way. And we found there's no significant difference between universities that don't plan on testing at all, did definitely plan on testing, or hadn't made a decision at the point of this database. So we're using this kind of cases per capita measure where we're using that case number I told you about earlier and dividing it by what the universities reported their enrollment was, again, prior to fall starting. So that number, this is very coarse data at this point, and hasn't been de-biased in any way. Yes. So we didn't see the planning have an effect on case numbers. However, perhaps more obviously, we did see an effect of whether or not they plan to have a hybrid model, an in-person model, or an online-only model. So we see that universities that plan to be entirely online had significantly fewer cases per capita than ones that had an in-person model, and then hybrid kind of falling in between the two. So we're glad that the data represents something that we expected to see there. Now, a surprising thing we've come across so far is that we're looking at universities that did have testing on campus in the fall. We divided them up into ones that had voluntary testing, weekly testing, and biweekly, so kind of course categories there. Some had intermediate frequencies. But what we noticed was that when we look at all cases, so starting from when the New York Times started collecting this data to the closest end date of the fall semester, we did see that there was a significant difference between cases for universities that only had voluntary testing and those that had mandatory biweekly testing. However, if we restrict to just what we know came from fall cases, we don't see a significant difference with test frequency. So some preliminary hypotheses that we have are that universities that had significant outbreaks in the spring were that somehow biases their decision whether or not to have testing at all or how frequently to have testing, and that maybe that's what we're picking up between the differences here. And of course, there's a large bias in whether or not a university decides to test weekly or biweekly or to have mandatory testing at all, and that's based on their budget. And so many universities couldn't even conceive of having that as a policy because it was so expensive. Yeah, so that's just a preview of some of the preliminary results we have from our limited data collection so far. I want to thank my two collaborators on this project, John Drake and Tierney O'Sullivan. And so Tierney is our statistician, and I'll say I did all the statistics on the preliminary results so far. So if you've noticed anything iffy there, I take full responsibility. And of course, thank you to MIDAS for supporting this work. I'm happy to answer any questions.
Great, thank you. Any questions? So I'll throw out a couple that come to mind. One thing that strikes me is you said a little bit about the interchange between on campus and off campus, but it would seem that the one possible hypothesis would be that the overall tenor of the policy in the surrounding communities could have had an impact on what happened on campus. Do you have any thoughts or comment on that?
So that's something we're hoping to get to in kind of a part 2 version of this, looking at how community-wide, like broader policies impacted the university, as well as university policies impacted the broader community. So, you know, the CDC released a MMWR report that kind of shows universities that chose to remain in person had significantly higher numbers of cases in the fall than those that went online, and of course that varied over time. So looking more deeply at that is something we want to do. Did that answer your question?
Yeah, it did. It did. I mean, it's a challenging question to get at, to model that in detail, I'd imagine. Since nobody else is speaking up, I'll throw out one other question. Did you look, or have you given any thought to some of the places that I believe were doing some of the non-individualized testing, like the wastewater testing?
Right.
And so that was going on at a few institutions.
Right. So we have to, based on looking at the plan database that we have, only a very few universities had the pooled testing. But I think that in the actual fall semester, more universities moved to that model. So we're meeting with Davidson College, who's been working on a much more accurate database as to what happened, and hopefully we'll get a better idea of who used pooled testing as opposed to individual based.
Yeah, great. Any further questions? Well, given that so many of us spend so much of our time on university campuses, I'm sure we're all going to be pretty interested to hear what you come up with on this. So thanks for your talk.
Thank you.
Okay, so our third presentation is from Keisuke Ejima, who's an assistant research scientist, and I apologize if I got your name wrong in any way there. He's an assistant research scientist at Indiana University, working on mathematical modeling of infectious disease and related topics. And so I'm looking forward to hearing what Dr. Ejima can tell us today. Take it away, please.
Okay. Thank you for kind introduction and giving me an opportunity to share my research idea and some results entitled Test-Based Guideline to End Isolation of Patients. I'm Keisuke Ejima, and I'm an assistant research scientist at Indiana University School of Public Health, Bloomington. And Marco Ageli is a associate professor in the same institute, and he's a co-I of this project. So massive vaccination campaign is ongoing in United States and elsewhere. However, it might take a while until the vaccine covers a sizable fraction of the population. And there remains some uncertainty around the vaccine efficacy against the emerging variants. So non-pharmaceutical interventions are needed to be used continuously. And a simple and effective NPI is isolating infected individuals to mitigate the transmission. However, lengthy isolation reduces the risk of transmission. However, it may be a burden for both patient and healthcare facilities. So guidelines for determining when to end isolation are needed to be needed considering the balance between cost of isolation and risk of transmission. So there are two main approaches have been adopted by many countries. One is to isolate patients for a fixed time period, which we call one-size-fits-all approach now. For example, the CDC's guideline for isolation include this statement. At least 10 days have passed since symptoms first appeared. However, this approach does not account for the individual variability in the duration of infectious period. So here's another approach. Another approach is based on the assessment of viral load of each isolated patient and isolation end when the viral load drops below a certain threshold value. Viral load is measured by real-time PCR. And for example, the CDC recommend using PCR testing in particular circumstances, such as for patients with severe immunodeficiency. The guideline include this statement. Results are negative from at least 2 consecutive repository specimens collected at least 24 hours apart. So here's the purpose of this study. One is to compare one-size-fits-all approach and personalized approach using the PCR test result. And the other is to define best practices for the use of PCR-based personalized approach. So here is a method. We developed a mathematical model of within-host viral dynamics. The model captures the typical dynamics of viral load over time, which typically exponentially increases at first and hit the peak and start declining. So it's typically create a bell-shaped curve. The model is parameterized by longitudinal viral load data. Based on the model, we perform simulations of viral dynamics and implement the 2 guidelines. The 2 guidelines are compared using the 2 metrics. One is the probability of ending isolation of patients who are still infectious. And the other is the length of unnecessarily prolonged isolation period. This is obvious, but ideal guidelines yield lower probability of premature ending isolation and shorter prolonged isolation period. And this is a little detail of the viral dynamics model. We assume 3 compartments for simplicity: uninfected cell, infected cells, and virus populations. Uninfected cells get infected by the virus and move to the infected cell population. Infected cells produce viruses with a constant rate. Infected cells removed by innate or adaptive immunity with a constant rate C here in this figure. Because the model captured the dynamics of acute infection, uninfected cells do not die or reproduce themselves because it's a relatively short time period. As we can only observe viral load B, the 3-dimensional model is reduced to 2-dimensional model using quasi steady state assumption. So the final model here, F, is the ratio between uninfected cells at time t and that at time 0. So it's defined as here. So here's the data we used. The model was parameterized using the longitudinal viral data of SARS-CoV-2 patient collected from 5 published papers. For consistency, the viral data were measured from upper respiratory specimens, so nasopharyngeal, nasal, nasopharyngeal, or oropharyngeal, pharyngeal, nasopharyngeal. So it's trying to make the data source consistent. And the data are from 3 papers are from Asian country and 2 papers are from European countries. We excluded patients who received antiviral treatment and for whom data were measured only once, 1 or 2 days, which is because 1 or 2 data points are not enough to estimate parameters, and the model does not account for antiviral treatment. So in total, we used the data from 30 patients. So here's a fitted curve for all 30 patients. But let's focus on these 3 patients for illustration purpose. So the dot are the data and the lines are fitted curves. As you can see, there is a large variation in the decay speed of viral load. So for example, these two patients, the virus persists longer than this patient. So there's a kind of variability in the viral dynamics. And this is why we thought the one-size-fits-all approach may not be ideal to reduce the risk of transmission and also may impose unnecessary long isolation on some patients. So we simulated viral load of 1,000 patients with measurement error and computed two metrics. The one is probability to prematurely and isolation, panel A, and the panel B is the length of prolonged isolation varying the period of isolation. Note that we assume the infectious disappears when the viral load drops below 10 to the third power copies per milliliter, which correspond to the blue line, this line, and this line. But it is still uncertain. There is some uncertainty on the infectious threshold, so we did the same simulation with by varying the infectiousness threshold. And under the one-size-fits-all approach, we changed the duration of isolation from 5 days to 50 days. So as you can see, the longer isolation period leads to lower probability of prematurely ending isolation. However, longer unnecessary prolonged isolation and a higher threshold lead to lower probability of prematurely ending isolation, however, longer unnecessary prolonged isolation. Including the CDC, some health authorities recommend 10 days of isolation since symptom onset, which correspond to this red line in each panel. And as you can see, probability of prematurely ending isolation was 54.4%, although the length of prolonged isolation was negligible. And to reach 5% or lower risk of prematurely ending isolation, so this point, isolation need to be continued for 23 days, which is kind of long. And the length of prolonged isolation becomes substantially long. In this simulation, it was 11.5 days. And we also did a similar simulation using the personalized approach using PCR test. In this approach, we can change two parameters, interval between tests and consecutive negative result needed to be observed to end isolation. So, and upper panel A, upper panel is the probability of prematurely ending isolation. So as you can see, the lower panel is the length of unnecessary prolonged isolation. So as you can see, combination of frequent tests and smaller number of consecutive negative results lead to higher probability of prematurely ending isolation. However, shorter prolonged isolation. And the area surrounded by dotted line correspond to those with 1% or 5% or lower risk of prematurely ending isolation. And triangle and circle in the lower panel correspond to the condition to realize the shortest prolonged isolation within each area. So it's a bit complex, but here is a summary of the simulation so far. When two consecutive negative test results repeated at an interval of 1 day, which correspond to CDC's guideline, the probability of prematurely ending isolation is 21.6%. And the length of prolonged isolation is negligible, 0.7 days. And to reach a 5% or lower risk of prematurely ending isolation, two consecutive negative results on PCR testing performed at a 3-day interval is necessary. And we further compared two guidelines under the same condition. So there are two conditions. Risk of premature ending isolation is below 5% or 1%. And we found quantitatively similar results from these two simulations, 5% or 1%. And for the personalized approach, the best combination of the number of consecutive negative test results and the interval of tests was selected for each infectious period. So as you can see, the personalized approach was not influenced by infectious threshold. We did the same simulation varying the infectious threshold, the three threshold. But as you can see, the personalized approach was not influenced by the threshold, and it led to shorter prolonged isolation compared with the one-size-fits-all approach. However, because the prolonged isolation for one-size-fits-all approach, so light blue bars, was influenced by infectious threshold values, the difference between the one-size-fits-all and personalized approach in prolonged isolation becomes smaller with higher infectious period. So here is a summary. When considering a fixed isolation period of 10 days for all patients, the probability of prematurely ending isolation was 54.4%. And when we use a personalized approach, 5% or lower risk of premature ending isolation can be achieved by requiring 2 consecutive negative results on PCR testing performed at a 3-day interval. And with a one-size-fits-all approach, the isolation should last 23 days, which is long, for 5% or lower risk of prematurely ending isolation, and it clearly imposing lengthy isolation phase. And here's the conclusion. The one-size-fits-all approach is highly sensitive to the infectiousness threshold, whereas the personalized approach, targeted approach is not. The personalized approach entails cost and logistic challenges because the multiple PCR tests should be implemented like 1-day interval or 3-day interval or whatever. So that is a kind of challenging part of this approach. Considering the emergence of new variants, a personalized approach could minimize prolonged isolation period without adjusting the guideline for new variant, each variant, because as you can see in this graph, the personalized approach directly measure the viral load. So it can be influenced by the threshold. And if the threshold is different between the original variant and new variant, even in that case, we can use a personalized approach. And here's a limitation and consequences. I'm sorry.
Please finish up.
Okay. So there are still ongoing studies. So here's some limitation and current direction. And I'd like to thank Marco Ageli, co-author of this project, and Yongdam Jeon, who is a PhD student at Kyushu University, who helped me with computation of this project, and the other co-collaborator of this project. And also, I'd like to thank the MIDAS Coordination Center for supporting this project. Thank you.
Great, thank you. Any questions? So I'm curious about one thing that comes to mind here. Is there any data on how long people actually isolate after having symptoms?
Well, I don't really know. I have to check the literature, but most of the guidelines say 10 days is enough to. Mm-hmm. Well, I don't know that why they say 10 days is enough, but people, most of the health authority use the 10 days of isolation.
Okay, and there's another question here in the question of being, when do you start the testing when you're doing personalized strategies?
In this simulation, I assume the test was started from the timing of symptom onset, but it might be not realistic because people do not show up to the hospital until they really feel sick. And the data says it may take 4 to 5 days until they visit the hospital.
Got it. Thank you. Any further questions? All right, great. Well, thank you, Dr. Ejima.
Thank you.
So our final talk is going to be from Chen, who is an assistant professor in public health sciences at UNC Charlotte. Dr. Chen works on the influence of individual spatial and temporal heterogeneity on pathogens, and looking how the interaction among these sources of heterogeneity influence transmission. Dr. Chen had a late-breaking family obligation that meant that he was not able to be with us in person. So what I'm going to do in just a second is to play a video that he recorded for the talk. So if you bear with me just 1 second here, we will get going. No, hold on. Share screen.
Morning, folks. My name is Chen. I'm an assistant professor in University of North Carolina at Charlotte. Today, I'm going to talk a little bit about my work on the content feature mining of COVID-19 social media posts and how it might help the model across different social cultural backgrounds. So the rationale of this study is that COVID-19 is a very complex social epidemiological system. And in addition to the clinical and epidemiological aspects,
Harry, I think you might need to unmute yourself so that we can hear the video.
social cultural background. And
Sorry.
it is very difficult.
Let's start over then. Sorry about that.
Morning, folks. My name is Chen. I'm an assistant professor in University of North Carolina at Charlotte. Today, I'm going to talk a little bit about my work on the content feature mining of COVID-19 social media posts and how it might help the model across different social cultural backgrounds. So the rationale of this study is that COVID-19 is a very complex social epidemiological system. And in addition to the clinical and epidemiological aspects, there are social, cultural, and political aspects involved substantially during this pandemic. There are two most important things that I think are especially important for model this pandemic. First, the government and health agencies' responses. The second is the public awareness, perception, and willingness to comply with these interventions. But the challenge of the modeling work is we lack of the data or the features, especially on the social, cultural, and political aspects, and how they might shape the disease dynamics of COVID-19. So in this study, I choose the social media data as an almost instantaneous source to comprehensively evaluate the public perceptions towards COVID-19. In this study, I used two social media platforms, the Sina Weibo, which is mostly used in China, and Twitter, which is mostly used in Western countries. Because there are tons of data social media posts, sampling is important to gain a comprehensive understanding. So in the initial step, I only use the daily viral posts with high number of shares, replies, and likes. In successive studies, I will expand this modeling framework to non-viral posts and try to understand how the general public react to COVID-19 on social media. So in order to extract features from social media posts, there are existing frameworks. So first is the LINQ, Linguistic Inquiry and Word Count. This has been developed in the early 21st century and is a very popular one. And a more current or more recent advancement is the deep learning-based feature extraction models, such as the Word2Vec or BERT. There are some problems with these existing feature extraction models. For instance, LINQ is almost exclusively developed in the Western social cultural background, and it is very difficult to apply LINQ into non-Western social media platforms, such as the social media in China. The deep learning-based extraction models, such as the Word2Vec, this one does not have a very good interpretability. It's based on deep neural network, so it's very hard to really understand what's behind the features. So to overcome these challenges, I propose a novel feature extraction scheme to first manually label the social media posts into multiple major and minor categories. These categories involve clinical and epidemiological aspects, countermeasures, policies and politics, public responses and social impact, spatial scales, and social problems. These features are interpretable comparing to deep learning based, because these are whenever a social media post has mentioned one of these categories, it will be labeled as 1, otherwise 0. So eventually it will generate a 0-1 binary vector for each social media post. The second advantage is this is universally designed, and this does not depend on a specific social cultural background. So when we talk about data-driven methods, the first and foremost challenge is how do we get a reasonably large number of labeled data. So in this initial stage, we have about thousands of viral posts labeled by our volunteer students, and we cross-check the intercodal reliability to ensure that the manual codings are accurate. Eventually, this manually labeled data will be fed into an algorithm to develop large-scale labeling system by supervised learning. This supervised learning can be rule-based or data-driven classifications. So I'm going to talk a little bit about what we get from this initial study on the data mining of social media posts in different platforms. As we can see here, the top 10 features on viral tweets and top 10 features on viral Sina Weibo posts are substantially different. This might reflect the different interest and emphasis of general public towards this unprecedented emerging pandemic. So for instance, in Twitter, public discussed mostly about the health agency, potential violence related to it, international issues, misinformation. On the Chinese Sina Weibo platform, people usually talk about the research, the transmission, the cases, health personnel, and testing. So this is very interesting, and we further did a dimensional reduction, and so we use TSME to see how the two social media platforms might differ. And as we can see here, the difference is quite substantial. The discussion content features in Twitter is quite different from that of Sina Weibo in China. And we further did a unsupervised clustering and further look at what topics are frequently concurrent and why. An interesting pattern we found that is, for instance, Bill Gates, vaccine, and conspiracy theory. These three features are almost always concurrent. So these are, on this slides, we can see that unsupervised clustering. We have the k-means clustering, and the pattern on Twitter and the Sina Weibo is again quite different. So this might explain the difference in the public responses and awareness on the social media, which can be a reflection of the compliance or the interest of the public. In the next step, these studies, the results in the previous slides are all based on the manual label. It's still quite intensive, so we want to apply a supervised learning or semi-supervised learning to accelerate this labeling process. We also want to perform a spatial temporal analysis to see how content features differ across time, space, and social cultural spectrum. Especially, what are the public sentiments towards interventions? So intervention is one specific type of content features in this study. Then, more importantly, we want to couple the online discussion features with the actual COVID-19 dynamics. We would like to use time series analysis and signal processing and identify the potential drivers of COVID-19 dynamics. And finally, we want to apply data-driven deep learning methods with transfer learning technique based on recurrent neural network to see if we can identify some hidden dynamics or hidden patterns buried by the social cultural factors. This is a quick presentation of my current work, and thank you for your time to join me, and thank you for MIDAS support with the grant. The Twitter data are provided by the UNC Charlotte School of Data Science, and I'm also supported by the Department of Public Health Sciences. The Weibo data is supported by my colleague from the Hong Kong Baptist University School of Journalism. I do have some related publications with this study, and if you like, you can take a look. If you have any questions, please feel free to email me, and my email address is on the first page of the slides. And once again, thank you very much, and I look forward to potential collaborations with you. Take care.
Okay, great. I don't know if Dr. Chen is on the line. He might have been, so, but I don't see him here. So I don't think we can take any questions, but please just get in touch with Dr. Chen if you have any further questions.
Yeah, hi. Actually, I'm online now.
Oh, you are on. Okay.
From my kids' school. Thank you so much for helping me with the
Got it. Thank you. Okay, so I'll ask a quick question. Do you see any trends over time in the data?
Yes, we actually, this is, I forgot to mention, this is the first 4 months. So we see some interesting trend and some very strong correlation between some, important events. For instance, when the WHO announces the PHEIC, the Public Health of International Concern, I think that was on January the 31st, 2020. It has a huge impact on the social media. And then on February, also think the WHO announced the pandemic. That's the second huge spike we can see. And that's on the Twitter side. For the Weibo side in China, we see this the lockdown of Wuhan. That was a huge impact, has a huge impact on the discussion trend, and also the ease, the lift of the ban of the lift of the lockdown has the second big impact, and we can see all different kinds of perceptions towards this countermeasures towards these policies. And then we will further analyze the sentiments towards them and see whether or not there can be any useful signals to drive the disease dynamics, or whether this disease dynamics actually trigger the public response. So that's our current observations, and we would dig into this more comprehensively.
Great. Well, thank you very much. So we're about out of time. So unless there are further questions,
sure.
I want to thank all of our presenters for their interesting work. I'm looking forward to hearing more strong results from the Urgent Grant Program. And thank you all for attending.